Hello students, I am Amruta Pimpelwadkar and I am working as an assistant professor in the Department of Biotechnology KTHM College, Nasik. In the previous session, we have started with a discussion about the topic antigen and antibody interaction. Now in this session, we are going to continue with our discussion and today we are going to discuss about some diagnostic tests which are used to detect the antigen and antibody interaction. So to revise, in the previous lecture, we have seen what do you mean by antigen, what do you mean by antibody, and how they are going to interact together to form the immune complexes. So the region on antigen which interacts with antibody is known as epitope or antigenic determinant, and the region on antibody molecule which is going to interact with the epitope is known as antigen binding site, or it is also known as paratope. Then we have seen how they interact with each other to form a visible precipitate, or this they undergo agglutination reaction to form large complexes and they separate out from the solution. The main phenomenon behind this or behind the interaction between antigen and antibody molecule is the lattice hypothesis that we have discussed, and then we have seen how there are three different zones in precipitin curve or agglutinin curve. These different zones indicate where actually the interaction between antigen and antibody will become visible. So we have seen that at the zone of equivalence, the concentration of antigen and antibody are nearly equal, or we can say they are optimum, and that's why precipitation or agglutination can be seen in this zone of equivalence only. So, in today's session, what we are going to discuss is, firstly, we will see what do you mean by a diagnostic test and what is its significance. Then we will see what is the basic difference between precipitation reaction and agglutination reaction. Basically, both of these are the interaction between antigen and antibody only, but there is one minute difference between these two that we are going to discuss today. Then after that, we will be discussing about the different types of the precipitation reaction. Now, what are our learning objectives behind this session? Firstly, we need to understand the significance of a diagnostic test. Now, when I am saying diagnostic, what I am referring to is a diagnosis part of the clinical microbiology, you can see, in under which we can detect the causative agent of a certain disease. So that is the significance of diagnosis, where we are interested in detecting what is the causative agent of a certain viral or bacterial disease, or we will say we want to detect the pathogen. Then secondly, we are going to learn the difference between precipitation and agglutination on the basis of type of antigen which is participating in that particular reaction. And after that, we will be discussing what are the different techniques which will come under these two reactions. Now, moving to our today's topic, firstly, we will see what is the test. So, diagnostic tests are also referred to as immunoassays. Now, assay refers to a specific protocol which is followed to determine either to detect the presence of a particular agent or to determine its concentration. And when we are saying that immunoassay we are going to perform, that means in this protocol or in this procedure, certain antigen or antibody molecule will be utilized. So to define, it is a technique or it is a test which is used to detect the presence or quantity of a substance such as protein based on its capacity to act as an antigen. Now, capacity to act as an antigen means I am referring to its property to bind with a particular antibody. Now, this can be qualitative also or quantitative also. Qualitative means what? If we are just detecting the presence of an antigen or antibody in the given sample, then it is referred to as qualitative, where we are not interested in its concentration. What we need to see just whether it is present or not. And in quantitative determination, what we are supposed to do is we are supposed to determine the concentration of antigen in the given sample or titer of the antibody in the given sample. So this is the difference between qualitative and quantitative. Now, if you see, there are different types of the diagnostic assays or immunoassays which are used for determining the presence as well as concentration of antigen and antibody. You can see this list of the immunoassays like precipitation reactions are there, agglutination are there, complement fixation test is there, ELISA techniques are used, then immunofluorescence, radioimmunoassay, and Western blotting. These all are the types of the immunoassays. From all these types, today we are going to focus on the precipitation reactions. Now, in last session, we have already discussed this. What is this? This is a precipitin curve or agglutinin curve. You can see in this curve that there are three different regions. Why I am emphasizing this again is because you need to be clear with the concept of zone of equivalence. So in this graph, what you can see is there are three different regions. On your extreme left, you can see that there is a prozone area, that is zone of antibody axis, and on the right side of this curve you can see that there is a zone of antigen axis which is called as post zone. Now you can see in this figure that in prozone or post zone area of this curve, there is either minimum precipitation, that is lattice formation, or there is no precipitation, that is there is no cross linking between antigen and antibody molecule. Now why this is so? Because in the first part there is excess antibody concentration present than the antigen, and in the third part there is there are lesser number of antibody molecules present as compared to the antigen. But in the middle of the graph, what is happening? Their concentration is becoming nearly equal. So you can see that there is proper cross linking between the antigen and antibody. Now proper cross linking means what? They are forming ordered complexes which is called as a lattice. So at the zone of equivalence, you can see maximum cross linking has occurred, and due to that what has happened is lattice formation has taken place, which can be seen as a visible precipitate, or in the case of agglutination reaction it can be seen to sediment at the bottom of the test tube or the plate which you are using to perform the diagnostic test. So here you need to remember that antigen and antibody cross linking will occur in the zone of equivalence where it will be detectable. Now, what is the difference between precipitation and agglutination reaction? Now, if you consider antigen, what are these? These are mostly proteins or glycoprotein molecules. Now, we have discussed the definition of antigen as it is any foreign molecule which can enter your body. If it is immunogen, then it can activate your immune system, and if your immune system is getting activated, that means B cells will start secreting the antibody molecules, and antigen can get attached to antibody. That is the main property of this molecule. Now here there are two types of the antigen. One is known as soluble antigen and second one is known as particulate antigen. Now these both types of the antigens are going to interact with the antibody molecule, but there is difference in their interaction. One basic difference is there. Now see, here you can see a figure. This is the case with the soluble antigen. Now when I'm saying that antigen is soluble, so what I'm referring to is, like for example, if you are adding 1 spoon of sugar into the water, what is going to happen? If you stir it continuously, then after certain time that sugar won't be visible. Why is it so? Because we see that sugar has got dissolved in the water. So this is the process of solubilization. Now in this figure you can see that red color circles are present, which are the antigen molecules, and you can see that they are dispersed in the solution. Now I'm saying that these particles are soluble in the given solution. So what will happen after certain time? If you keep on stirring them, then what is going to happen? They will get dissolved. That means they will disappear in the solution. Why this is so? Because they are forming uh, they are interacting with the water molecules or the solvent molecules which are present in the surrounding. So this is the meaning of soluble antigen, that means which is miscible in the given solvent. So now if I am considering this soluble antigen, what will happen when I am adding antibodies in this solution? Now I have added known antibodies which are specific against the given antigen. So what will happen? Antibodies and antigen are specific for each other. So what is expected? They should interact with each other. Now when they are interacting with each other, what they will do is they will form cross linking, and hence here what is happening, before this antigen was present in the dissolved form, that is in the soluble form, but now due to the presence of antibody molecule, what has happened is this antibodies have cross linked the given antigen molecules, and due to that they have converted this insoluble antigens, soluble antigens into the insoluble immune complexes. So here what has happened is soluble is converted into insoluble immune complex, means now it will be visible as a precipitate, and these antibody molecules which are causing the precipitation of the given antigen, given soluble antigen, are referred to as precipitin molecules. Now what will happen with this immune complexes is they will sediment at the bottom as a insoluble precipitate. This is the case with soluble antigen. Now, what happens with the particulate antigen? First thing is, what do you mean by particulate antigen? Particulate means a molecule which cannot be dissolved in the given solvent, or which is not soluble in the given solvent. So here, as I'm referring to an antigen, so what will happen is this antigen cannot be dissolved in the water, or it won't get dissolved in the blood sample. So like for example, if you are taking sand and you are adding it into the water, then even if you keep on stirring that solution, after certain time also that sand will remain as it is and it won't get dissolved in the water. So same way, this particulate antigens will remain in the solution, but they will not get disappeared, that is they will not get dissolved. So when you are adding antibodies in such solution, what will happen is these antibodies will form cross linking between these particulate antigens, and they will form larger complexes. Now such antibodies which are interacting with the particulate antigen are referred to as agglutinates. Why? Because they are performing agglutination reaction. Now what do you mean by agglutination? Where larger complexes are formed by combining smaller units together. That reaction is called as agglutination reaction, and that's why these antibodies are referred to as agglutinin. So what will happen here, as the size of complex has increased, now it is going to sediment at the bottom due to the gravitational force. So this is the basic difference between precipitation and agglutination. In precipitation, what is happening? Soluble antigen is interacting with the antibody molecule, and that soluble antigen is converted into insoluble precipitate in the form of an immune complex. And in the agglutination, what is happening? Your antigen is already particulate, just it is linked together to form larger clumps which will become visible. That is the agglutination reaction. So particulate antigen will combine with the antibody molecule to perform agglutination reaction. Now today in this session, we are going to discuss about the types of the precipitation reaction, and we will continue this discussion in the next session to consider the agglutination reactions. So today as we are going to discuss about the precipitation reaction, this can be performed in two different mediums. Either they can be performed in fluids or they can be performed in gels. In fluid, when this reaction is performed, it is called as precipitin ring test. We are going to see how it is performed. And in gel, when it is performed, there are two types again. One is known as immunodiffusion, and if it is combined with the technique of electrophoresis, then it is referred to as immunoelectrophoresis. So now we will discuss about the precipitation reaction in fluid. So as I have told you, this test is referred to as precipitin ring test. Now why this is performed? This can be performed as a qualitative technique, that is just to see whether antigen or antibody is present in the given sample or not. If you are doing it qualitatively, then you just need to detect the presence of precipitate. But this technique can be modified to determine the antibody titer. That means the concentration of antibody which is present in the given sample. So we are going to discuss this modification today, how this test is performed now. For performing this test, you need to take some test tubes, empty test tubes, and in the first step, what you need to do is you need to add equal volume, that is same volume of the antigen with known concentration. Now, when you will take the antigen of known concentration, if you want to determine the antibody titer in the patient serum, then you will take the antigen of known concentration and you will add it in equal volume in all the given test tubes. Now, next step is the addition of glycerol. That means what you will do is you will add a layer of glycerol on all the given tubes. Now, what is the purpose of adding glycerol? It will prevent the mixing of the two solutions which you are adding, plus it is going to provide you one interface at which antigen and antibody can diffuse and interact with each other. The next step, that is third step of this test, is addition of the serum sample. Now, why you are taking serum sample? Because you want to determine the concentration of antibodies present in the patient's serum. So here you need to add the serum sample. But how you are going to add that now? In the first tube, you are going to add the original serum sample which is having specific concentration of antibodies, that is antibody titer. Then what you need to do is you need to perform twofold dilution of this serum. Now, what do you mean by twofold dilution? Like you can see in this figure, on first tube you can see the label as 1. That means it represents original serum sample. And on the second tube you can see the label that is 1:2. That means half. So 1:2 dilution is performed means what? In this figure, for representation I have given 40 triangles, that is triangles are representing the antibody molecules here. So I have indicated I have given 40 triangles in the first tubes. That means 40 antibody molecules are present originally in the given serum sample. Then in the second tube what you can see is that number is minimized. Minimized means what? It is reduced to half. Only 20 molecules you can see which are present in the second tube. In the third tube, 10 molecules are present. So it is 1:4 dilution. So what is happening in the twofold dilution? The amount of antibodies which is present in the original serum sample is getting reduced. So what will happen here? We have kept the antigen concentration constant, that is same in all the tubes, but what we are varying the antibody concentration. So recollect your knowledge about the precipitin curve. There same thing was happening, one molecule was kept constant and second molecule was the concentration of second molecule was varied. So here now what is going to happen? In first tube, now you can see that there are more number of antibody molecules present as compared to the antigen. Antigen means a red colored circle. So here in the first tube, if antibody molecules are present in excess concentration, that means which zone it is forming? It is forming a zone of antibody excess. And if you see last tube now, that is the one having 1:16 dilution. Till this point antibodies have got diluted, and due to that now in this tube you can see antigen is constant, so number of antigen molecules have increased. So in the last tube what has happened, as compared to antibody, antigen is present in more concentration, so it is forming the zone of antigen excess. So in both of these tubes what you can see is there is either antibody excess or antigen excess, so there won't be any precipitation in these two tubes. But what will happen in the middle three tubes, that we will see now. After this you can observe for the precipitation. Now where precipitation can be observed? In the layer of glycerol. Why? Because antigen from the lower layer and antibody from the upper layer are going to diffuse in the glycerol layer, and there they are going to interact with each other. So in the middle three tubes, there is nearly zone of equivalence which is formed. That means antigen and antibody molecules have attained a concentration which is nearly optimum, that is nearly equal. So at that point you can see a visible precipitin ring which is formed, that is indicated here with a black line. So more precipitate is formed in the 1 is to 4 dilution. Why this is so? Because in that tube you can see 10 molecules of antigen and 10 molecules of antibody are present. That means they are near to the equal. So here in this tube, zone of equivalence can be seen and their maximum precipitation can be seen. On the either side of that tube, you can see precipitation is decreased because either it is going to the zone of antigen excess or it is going to the zone of antibody excess. So this is the basic principle of the precipitin ring test, and by performing such dilutions you can find out the antibody titer. Antibody titer is the highest dilution which is showing you positive precipitation. That means it is the concentration of antibody molecules which is required for performing the precipitation. So here you can see highest dilution which is giving you positive precipitation is 1 is to 8. So for this serum sample, 8 will be the antibody titer. So this is how precipitation reaction can be performed in fluid to determine the antibody titer. That is quantitative. Then next is precipitation reactions can be performed in gel. Now in gel what will happen is, gel means I am referring to an agar matrix. So you all know in electrophoresis also we use agarose slab or agar matrix. In that what happens is it is a porous matrix through which molecules can get diffused. So same way in this also, when we are using agar matrix as a medium, in that antigen and antibody can get diffused, and at a particular point they will come in contact with each other, and there they are going to form the immune complexes which will be visible to us as a precipitate in the gel slab. So here as antigen and antibody are getting diffused through the matrix, this process is referred as immunodiffusion. Now again there are two types of the immunodiffusion techniques. One is referred to as double immunodiffusion, which is discovered by Octorloni, and hence the method is given a name as Octorloni's method. And second is the radial immunodiffusion, which is developed by the Mancini, so it is referred to as Mancini's method. Now one by one we will discuss how these two techniques are performed. So first technique is double immunodiffusion. Now why it is referred as double? Because both antigen and antibody will get diffused. That's why it is called as double immunodiffusion. Now how it is performed? What you need to do in this technique is you need to take one agar slab. This gray colored rectangle that you can see on screen represents one agar slab. In this, you need to make two wells by using gel puncture. Like here, you can see the distance between these two wells should be minimum. Actually, it depends on the concentration of antigen and antibody present in the sample. So, these two wells should be prepared. Now, in the first well, you are going to add the antigen, and in second, you are going to add the antibody. Remember that red colored circles are the antigen molecules and green colored triangles are the antibody molecules. So when I am adding these two solutions in these two wells, what is going to happen? In the well, concentration of this particular molecules is higher, and in gel slab there are no molecules of antigen or antibody present. So what is the role or what is the rule of diffusion? I will say that the molecule will get diffused from its higher concentration to the lower concentration. So what will happen here? These molecules will get diffused in all the direction which is available for them. But what will happen in the middle part, that is between the two wells? Now consider the antigen well. What is happening in the well? Concentration of antigen is higher. It will go on decreasing as the antigen molecules get diffused towards the antibody well. Same way, if you consider the antibody well, then in the well concentration of antibody is higher and it will go on decreasing as antibodies diffuse towards the antigen. So now here what is happening, both these molecules have got diffused to the agar matrix, but at the middle part what is happening, see, this three zones are developed now. Near the antigen well, antigen concentration is more, antibody concentration is less. So that is forming the zone of antigen axis on the left side. Then on the right, near antibody well, what is happening? Antibodies are present in more concentration and antigen is present in lower concentration. So there zone of antibody excess is formed. And what is happening in the middle part? You can see their concentration has become optimum. So precipitation will not get inhibited in this part because it is forming zone of equivalence, and due to that you can see that precipitate will develop in this part only, that is in the middle of the two wells. That precipitate can be visible to our eyes. So this black color light indicates the presence of precipitate. This is the actual image that shows you how these results will be visible. So you can see there is agar matrix and two wells are prepared. In these two wells, antigen and antibody samples were added, they got diffused, and in the middle of these two wells they have formed a precipitin line which is visible as a white precipitate. So this is how double immunodiffusion is performed. Now this technique can be used to see whether the given antigen and antibody sample are specific towards each other or not. Means what? You can do qualitative detection. So what you can do is, like for example, I want to see whether a given bacteria is present in the water sample or not. Then what I can do is I can take antibodies against the proteins which are present on the given bacteria or given virus. Like for example, if virus is considered, then if you want to detect the influenza presence of influenza virus in the given sample, then what you can do is you can take antibodies against hemagglutinin or neuraminidase. Hemagglutinin and neuraminidase are present as proteins on the surface of influenza virus. So what will happen if these antibodies interact with the given sample? That means if virus is present, then precipitate will get formed. But if in the given sample virus is not present, then antibodies are not going to interact. So in that case, you won't be able to see any precipitin line here. So qualitatively, if you want to detect the presence of any protein or antigen in the given sample or a pathogen, then what you can do is you can take antibodies against that antigen, and then if precipitate is formed, that means antigen is present. If precipitate is not formed, that means antigen is absent. So this is one simple technique that can be used. Now, this technique can be modified to see whether two antigens are identical or not. That means to determine the pattern of identity. Now, what do you mean by this? We have seen if any antigen is considered, then it is going to have multiple epitopes on its surface. Antigens are polyvalent. That means they bear multiple number of repetitive epitopes on their surface. Now, in this image, what you can see, there are two types of antigens. One is A, second one is AB. Now why they are named so? You can see A is having red colored circle on its surface. That means it is having one particular type of epitope on its surface. Now if you see second antigen, that is AB, then it is bearing two types of epitopes on its surface. One is red colored, second one is yellow colored or orange colored triangle. So what is happening here? These two antigens are sharing one epitope, and one epitope is different on the second antigen. So here what you can say is these two antigens are partially similar. Why? Because if I am adding one antibody which is specific against this red circle, then what will happen? This red circle epitope, then what will happen? That antibody can interact with both of these antigens because that epitope is present on both the types of the antigens. So in this case, these two antigens are similar. How I have detected that? By using same antibody molecule. So to see whether to perform this test, you need to have two samples of antigens and you need to have polyclonal antibodies. And if same antibody is interacting with the two antigens, then you can see that these two antigens are bearing similar epitope on their surface. If this antibody is forming different lines, then you can see that these two antigens are not identical. How it can be detected, that we are going to see now. Now first is how pattern of identity can be seen. Now what you need to do here is, this is double immunodiffusion technique only, it is the modification of this technique. So what will happen in this, antigen and antibody both will get diffused to the agar matrix. So you need to take one agar slab. On that you need to make three wells like this. Now this first well you can see in which antibody is added, and in remaining two wells you are going to add the antigen sample. Like this only you need to make well in the shape of a triangle. Now, if you consider these two antigens, then in this case, what is happening? In both the samples, same type of antigen is present. If same antigen is present, that means what? It is bearing the same epitopes on its surface. Now, what will happen here? All these three will get diffused in the given agar matrix, and after diffusion, what is going to happen? If these antibodies are specific against the antigen in first well, then there it is going to form a line of precipitation. Then if it is specific against the second antigen also, then again precipitin line will be formed between the two wells. Now, after this, what will happen? Now, what I have told you, these two antigens are bearing similar epitopes on their surface. So, what will happen here is, these two precipitin lines can form cross linking with each other, and so they will get fused together to form one arc like this. So, if in this test such an arc is formed, that indicates that two antigens are identical. Means what? How you will detect the pattern of identity? If line of arc is seen between these three wells, then that indicates that the given antigen samples have similar epitope on their surface, and they can be referred to as immunologically identical. Now second is pattern of non-identity. Now, what do you mean by this? Again, by similar way, you are going to prepare three wells in the triangular shape, and you are going to add the given antigen samples. Now, in this case, what has happened? There are two different antigens you can see in this figure. One is A and second one is B. Now, in this case, both are having different epitopes. That means they are not sharing any similar epitope on the surface. They are totally different. So what will happen when I am adding antibody solution in the given well? All these three will get diffused and they will come in contact with each other between two wells. So what will happen with the A antigen now? Antibodies are specific against it, so it is going to form a precipitin line. Again, I have added polyclonal antibodies, so that will be specific against the second antigen also. So there also precipitin line will be formed. But what is happening now? These two are totally different antigens. So what is happening? These two precipitin lines won't get fused with each other. What will happen is they will remain separate, and they will form a cross like shape, as you can see in this figure, that a cross is formed of the two precipitin lines. So when such cross is formed, means what? These two lines have crossed each other and they have not got fused because they are not having similar epitopes to share with each other. So such type of pattern refers to two different types of antigens. That means two antigens are not identical with each other. That means they are not sharing the same epitope. So if you observe such cross, then you can interpret it as pattern of non-identity. And third pattern is of partial identity. That means what? Again, same way you are going to perform this procedure, but what will happen in this case now? You can see again two antigens are there. In the first, again A antigen is present in the first well, and in the second you can see AB antigen is present. So similar to our blood group epitope, what will happen? A blood group will carry A epitope on its surface, and AB blood group will carry both, that is A as well as B. So what is happening in the case of these two antigens now? They are having one epitope which is same, that is A type. But second antigen is carrying one more epitope, that is B, which is not same. So when I will add antibody now, again they are going to get diffused to the agar matrix. Now what will happen? These antibodies are specific against the A epitope, A type of epitope. So there it is going to form one precipitin line. Again, this antibody, second antigen is also having A type of epitope. So it is also going to form the precipitin line. Now what will happen? These two lines are of similar epitope or identical epitope. So they are going to get fused. But what is happening with the second uh, second antigen type now? It is having one more epitope which is going to interact with the polyclonal antibodies. But as it is not same as with the A, it is going to form a different line which will be seen as a spur. So you can see this pattern. So if this kind of pattern is seen, that is one line is fusing and second line is crossing, then this pattern is referred to as pattern of partial identity. That means two antigens are sharing some epitopes, but they are having unique epitopes too. That means they are partially identical to each other. So this is how double immunodiffusion technique can be modified to detect whether two antigens are similar or not. In this image, you can see how these three different patterns are visible. First one is the pattern of identity showing the fusion of two precipitin lines. Second one is pattern of partial identity where you can see one line is fused and second line is crossing. And third is the pattern of non-identity. That means two precipitin lines will be crossing each other and they will form a cross-like shape. So this is all about the precipitation reaction in gel, that is double immunodiffusion. Now second type of precipitation reaction in gel is radial immunodiffusion, that is Mancini's method. Now why it is referred to as radial, that you will know when we will see the shape of the precipitate which is formed. Now again in this technique you need to have the agar matrix, because what is happening in the immunodiffusion, antigen and antibody are getting diffused in the agar matrix. But what is the difference between double and radial immunodiffusion now? When you prepare agar matrix for this radial immunodiffusion, what you do is when you dissolve agarose in buffer, at that time only when it cools to the 40 to 45 degrees Celsius, you are going to add antibody solution. You will mix it properly and then you are going to load it in the tray. So what will happen here is you will get agar slab in which antibody molecules are uniformly distributed, like you can see in this figure. So what is the difference between double and radial immunodiffusion? In radial, you are adding antibody solution in the well. In the radial immunodiffusion, antibodies are added in the agar slab only. That means they are uniformly distributed throughout the agar slab. You are not going to add it in the well. So here you need to prepare only one well, that is for the antigen. So you will make one well and you will add the antigen sample in that well. Now what is happening in this case? Antibodies are already loaded in the agar matrix, but you are adding antigen inside the well. So who is going to diffuse? It is the antigen molecule. So antigen molecules will get diffused in this agar slab in all the directions. After diffusion, what will happen? They will be distributed. Now again, what will happen is antigen concentration near the well will be higher, and as it goes on diffusing, its concentration is going to decrease. In all the directions, it is going to decrease. But if you consider antibody molecules now, then all over their concentration will be equal. So this is indicated with the green rectangle you can see here. So what is happening in this case now? Antibody concentration is remaining constant and antigen concentration is changing. So what will happen at a particular point? There will be zone of equivalence which is formed. Okay, near the well there is zone of antigen excess, and far from the well there is a zone of antibody excess, but in the middle region you can see that concentration of both of concentration of this two is becoming optimum, and due to that zone of equivalence is attained, and this is seen as a precipitin ring. As you can see precipitation in the shape of a ring, this technique is referred to as radial immunodiffusion, and you can see the black color circle. This is the precipitin ring which is formed. Actually, how it is visible, you can see this image. Now in this, what is done, you can see that one plate is there, multiple wells are prepared, and in that multiple antigen solutions are added. Now all these antigens were having different concentrations, and they have given rise to the formation of precipitin ring. But what happens in this case is, when precipitation is observed, that means qualitatively you have detected the presence of an antigen, but this technique can be modified to determine the concentration of antigen which is present in the given sample. For that, similar to the biochemistry practicals, we need to perform the preparation of a standard graph. Now you can see the size of a ring. So the diameter of this ring or area which is covered by this precipitin ring is proportional to the concentration of the antigen. That means lower the concentration of antigen in the given sample, lower will be the diameter of this precipitin ring. Higher the concentration of the antigen, higher will be the concentration of the antigen in the given sample. So now for quantitative determination of antigen in the given sample, what you need to do is you need to take some standard solutions of the given antigen, and you need to add them in the well. Along with them, one well you are going to use for your unknown solution. Then what will happen is, as antigen concentration goes on increasing in the in the uh, known antigen solutions, the diameter of this ring is going to increase, and you will plot one graph like this. What you can see in this graph is, on the x-axis concentration of antigen is given, and on the y-axis you can see zone of precipitation, that is diameter of the precipitin ring is given. So in this figure what is given, 1, 2, 3, 4 are the wells in which antigen solution was added, and in this direction only concentration of antigen was increasing. So as the concentration of antigen has got increased, the diameter of the ring has also increased, and that can be seen as a linear graph. Why? Because they are directly proportional to each other. Now in one well you have added unknown solution also, so you are going to have a diameter of unknown with you, so that you can extrapolate on this graph. You can see the diameter of the unknown concentration is extrapolated on this graph, and then it is extrapolated on the x-axis. So by using this technique, what you are doing by extrapolating these values, you are detecting the concentration of antigen which is present in the unknown solution. So like this, by preparing standard graph and by running the unknown concentration, what you can do is you can modify this technique to perform the determination of antigen concentration in the given sample. So this is how radial immunodiffusion can be used qualitatively as well as quantitatively, and this is all about the precipitation reaction in gel. Now in today's session, what we have seen first is, we have seen that diagnostic tests allow us to detect and quantify the antigen and antibody in the test sample by doing just simple modifications in the protocol. Then after that we have seen that there are two types of the antigen, and hence there are two types of antigen antibody interaction, that is precipitation and agglutination. And after that we have seen the significance of precipitation reactions, that is how they help us in detecting the presence of antigen or antibody, and how they can be modified to determine their concentration also. In the next lecture, we will be discussing about the immunoelectrophoresis technique under the precipitation reaction, and then we will be discussing about the remaining types of the immunoassays. Thank you.